Okay, welcome everybody to Chapter 4, and what we're going to discuss today is the next level of organization in living systems, which are tissues. Essentially what tissues are teams of cells that have a similar structure and function that act to perform a particular job in the human body. And so histology is defined as the study of tissues. Tissue classification is based on cell structure and the composition of the surrounding environment near the cell, which is called the extracellular matrix, as well as a study of cell function. And there are four main cell types that we're going to address today: epithelial, connective, muscle, and nervous tissue. And even though they all have the same DNA in their nucleus, they look and act very differently from each other because they perform different tasks. Histology is the microscopic study of tissues, and it involves a variety of techniques: sectioning, staining, and essentially an analysis of cell shape and cell function. When we perform a biopsy, we actually remove living tissue for diagnostic purposes, and what we're trying to do often in a biopsy is detect perhaps the presence of a pathogen or perhaps a change in cell structure or function that could indicate some other type of disease state. In an autopsy, we examine the organs of a deceased individual and we attempt to determine the cause of death, um, in part by doing histological study, in part by doing toxicology, where we look at the levels of certain chemicals in the cells and in the system, such as in the circulatory system, the lymphatic system, in the extracellular fluid, and so forth. Okay, one of the things that we need to discuss before we get into the histology of the human organism in its adult stage is where these tissues come from. And what we have to understand about human development is that it all begins at fertilization, and there's a very important event that happens shortly thereafter where we form what we call the three germ layers, and these are the endoderm, the mesoderm, and the ectoderm. Now, let's first try and describe what happens when the sperm meets the egg. Okay, so we have fertilization, we have the sperm, we have the egg here, and they'll fuse at fertilization and they'll form a cell called a zygote. This is a diploid cell, contains 46 chromosomes, and what will immediately happen is that this fertilized egg, the zygote, will begin to rapidly divide and form a cluster of cells that looks a little bit like a raspberry, and what we call this is a morula. Okay, so pardon my art here, I'm drawing with my fingers, but bottom line, morula. And what will happen is that this cluster of cells will become larger and larger, and it will move down the fallopian tube away from the ovary and towards the uterus. And at some point what will happen is a process called hatching. And in the process of hatching, what will happen is that this solid ball of cells will form a hollow ball of cells called a blastula. Okay, blastula. Now, let's make what I would consider to be a coronal section through the blastula, and what you'll see is that it is a hollow ball of cells that's fluid filled, and on one part of this fluid filled ball is attached a clump of cells called the inner cell mass, and that will eventually be the embryo. Okay, so we'll indicate the inner cell mass in red here. And there's the rest of this fluid-filled ball, just liquid in there. What will happen next is it will undergo a few different changes, and these changes include morphogenetic movement. That's basically cells moving from one place to another inside this structure, and differentiation, where cells actually take on different characteristics in terms of their function and their structure. And what will happen is that in the context of the inner cell mass and this outer layer, this blastocyst, what we'll eventually get to is a structure that, if we were to take a coronal section through it, would look a bit like a flattened disc. So let's sort of draw this here. Okay. And you might imagine, well, how do we get to this flattened disc? Essentially, what happens with the blastula is that certain populations of cells divide more rapidly than others, and what this forms now is this flattened structure with an indentation on one side. Okay, so here is a cross section through the embryonic disc. Okay, and again, this is all lined with cells, and there's fluid in between these two cell layers, and these two cell layers at this point have begun to change to the point where the cell layers that are on the top we call the ectoderm, and the cell layers that are on the bottom we call the endoderm. Okay, so we're kind of drawing this here. Okay, and this is all action that's happening in a certain location within this inner cell mass. Okay, so we're kind of zooming in on this portion of the blastula here, and sort of walking up and taking a close look. And so what happens is you get these ectodermal cells and these endodermal cells, and they start to separate a bit from each other. And so this would be the ectoderm here, and this would be the endoderm down here. And then what will happen next is that through the magic of morphogenetic movement, a small crease will develop inside this embryonic disc. And so what I want to do is kind of redraw this real quick for you. So let's just go back to the pen here. So just stay with me. Here's the embryonic disc, and what will happen is that a tiny groove will develop in the top called the primitive streak. And for those of you who want to kind of get oriented, the ectodermal layer represents what will eventually be the dorsal surface of the embryo, and the endodermal layer is eventually going to be portions of the lining of the gut and basically a lot of the structures of the internal organs. So what will happen now is that cells that are in this ectodermal layer will begin to migrate through this primitive streak through the magic, again, of interaction of cell receptors with certain molecules that are secreted by the cells around them that tell them essentially where to go, kind of like a chemical breadcrumb trail. And certain populations of cells, and we'll indicate them in red here, will migrate through this primitive streak and start to populate this opening that exists between the ectoderm and the endoderm. And what these cells will become are mesodermal cells. Okay? Now the reason that this is a major event in development is because once we reach this point, the cells can't go back in time and be something else. A mesodermal cell can't go back and become an ectodermal or an endodermal cell. An ectodermal cell can't become an endodermal cell. Basically, their fates have been restricted as a result of changing what portions of the DNA those cells are allowed to read from, and as a result, changing what kind of protein products they produce, which is in turn going to change their structure and their function. Okay, so that right there is the mesodermal layer. Okay? And so what do each of these different germ layers form? Well, the endoderm is going to form the lining of the digestive tracts and the digestive tract derivatives, structures such as the pancreas and the liver and so forth. The mesoderm is going to form the musculoskeletal system and all of its components, and a lot of the cardiopulmonary system. And the ectoderm is going to form the epidermis, the skin, and the nervous system. Okay, so that's a bit of an oversimplification, but that's basically where the adult structures are going to come from. What will eventually happen is that this disc will begin to slowly change shape. Eventually, it'll establish a superior-inferior axis down the midline, which will become visible as structures called somites. That will eventually lead to the formation of the spinal cord and the brain, and those will eventually be enclosed by the skull and the vertebral column. When we establish that longitudinal axis, we'll also establish bilateral symmetry, so the embryo will have a left and a right. We'll then begin to see vestiges of the eyes and the ears and the upper and lower limbs. At one point, there will be a tail that will eventually resorb and become nothing but a vestigial structure called the coccyx. And then differentiation will continue and growth will continue until we go from the embryonic stage to the fetal stage to the neonatal stage. And all this time, what's happening is that these germlines are giving rise to more and more differentiated cell types that are going to perform a particular function in the adult organism. Okay? So that's the bottom line. That's where these different terms come from. I just wanted to kind of give you a look ahead into one of the later chapters in the book so that you could understand what we mean when we talk about the germ layers and why they're significant. So the first tissue we want to address is epithelial tissue. And in general, one of the ways to distinguish epithelial tissue from most other tissue types is that the cells in these tissues are very closely connected to each other, and there's not a lot of extracellular space between them. That's one of the ways that you can differentiate, for instance, epithelial tissue from another tissue type called connective tissue. There are some other characteristics that are germane to nervous tissue and muscular tissue that we'll get to when we actually reach that part of the talk. So what does epithelial tissue do? It covers body surfaces, it forms glands, it's found on the outside surface of the body where it's an important portion of the cutaneous membrane, which is our barrier between the outside and the inside world, so this prevents infection. This is also one of the body's major sources of vitamin D. It's a thermoregulatory organ. It regulates our fluid balance, and it's a major sensory organ as well. Okay. It also lines the digestive, the respiratory, and the urogenital systems. And what it does there is it serves as a nice barrier, keeping stuff inside the organ inside it and the stuff outside the organ outside it, and it only allows certain things to pass through this barrier, and that's controlled by the transport processes in the cells that make up these epithelial linings. There's also a lot of secreting that goes on in a lot of epithelial coverings, where we will make a cellular product and then we'll shoot it into the external environment, and it'll perform a particular function for the organism. As an example, if you look at the upper respiratory, it's lined with a special type of epithelial tissue called pseudostratified ciliated columnar epithelium, and it produces mucus. There are specialized cells called goblet cells that make mucus, and what this does is it helps trap particulates in the air that we breathe so that they don't end up down in the lungs. Okay, so that's an example of a secretory function of epithelial tissue. We also find it surrounding the heart and the blood vessels and lining a lot of different body cavities. It's a major component of a lot of membranes in the body. It has a free, a basal, and a lateral surface. The free surface is often called the apical end, and it's often populated by specialized structures such as microvilli, and or cilia. It has a basal surface, which is generally attached to a membrane called the basal membrane, which is an extracellular secretion of the epithelial tissue and serves as an anchoring point to connect the epithelial tissue to the underlying connective tissue. And there's lateral surfaces, and those are generally bounded by other epithelial cells. The basement membrane, again, is an extracellular secretion that allows the epithelial tissue to adhere to other tissue types, mostly connective tissue. And there are also specialized cell junctions that we'll talk about a little bit later that perform a variety of functions. Some of them resist mechanical stress. Some act as barriers so that substances can't pass between epithelial cells. And some act as communication networks to allow certain substances to move very easily from one cell to the next. They tend to be avascular, meaning there are not a lot of blood vessels running through epithelial tissue. Most epithelial tissue gets its nutrients and its fuel as a result of diffusion due to its proximity to specialized blood vessels that are called capillaries, which themselves are very thin-walled. As we'll see when we get to the section on stratified squamous epithelium, one of the things that happens as epithelial cells lose contact or basically increase their distance between the blood vessels and their diffusible substances, they begin to be unable to maintain metabolism because they can't get oxygen and nutrients, and the cells will slowly begin to die. And in stratified squamous epithelium, that's by design. They also have an amazing power to regenerate. They have a high mitotic index, and the reason for that is that a lot of epithelial tissue is exposed to harsh environments, and as a result of cell damage, frequently cells die and have to be replaced. And if this didn't happen, we'd rapidly lose our epithelial covering. Now, the basement membrane is a matrix that's formed by secretions of the epithelium and the connective tissue, and it's a glue that allows attachment to underlying connective tissue, which itself is a type of glue. It guides cell migration during tissue repair, so it acts, again, as a type of system of chemical breadcrumbs that direct cells to the proper location in the event of injury. And it acts as a filter in the nephrons of the kidney. Now, we haven't talked extensively about the kidneys yet, but one of the kidney's jobs is to clean your blood, and in order to do that, we have to let certain substances stay in the blood and let other substances pass through in order to remove waste products and retain stuff that's critical for our survival. And one of the membrane layers in the nephron of the kidney is the basement membrane that lies between the capillaries that are part of the nephron, which are specialized for this kind of filtration. They're called glomeruli, and between the initial part of the kidney tubule that catches the filtrate, which is called the Bowman's capsule. And so there's actually a series of filtration membranes that these substances have to go through in order to be part of what will be deemed the filtrate and what will ultimately become the urine, and a portion of that is generated by the basement membrane between the capillaries and the surrounding tissues. Not every epithelium is going to have a basement membrane associated with it. An example of one that does not are the hepatic endothelial cells that line the sinusoids of the liver. The liver is an amazing organ. It does so many things that we literally couldn't exist without it. That's why it has as its prefix the word LIVE, to live, because it's that critical to our survival. What happens with the liver is that it receives blood that's drained from the digestive system and the spleen, and it's responsible for detoxifying the blood, making plasma proteins, checking the blood for the presence of pathogens, and storing nutrients that are in the blood for later use in the case of carbohydrates and fats. What happens is that the liver has the ability to store them as adipose tissue or as glycogen, and in the case of protein, it can sometimes store that as fuel for later use, or depending again on the metabolic needs of the body, the liver may allow those nutrients to pass directly onto cells so that they can be used for metabolism. So that's an exception to the rule that there's a basement membrane associated with every epithelial tissue. Now, once that blood has passed through the liver, what happens is that it eventually is going to collect in central veins that run through systems of liver cells, which are called hepatocytes, and that will eventually drain into the hepatic vein, which will eventually drain into the inferior vena cava, and that leads directly back up to the heart. And so by that time, the blood has been essentially cleaned and regenerated. Okay, so what are some more functions of epithelial tissue? Well, it protects underlying structures, such as the lining of the mouth, where it resists mechanical stress so that it doesn't tear. So this is a protection against infection. It acts as a barrier against the outside world. We've already discussed the skin's role in preventing infection by serving as a barrier, and it permits for passage of substances through it, such as the nephrons in the kidney, or if we look at the small intestine, the ciliated columnar, the brush border and the columnar epithelium associated with that line the small intestine that allow passage of nutrients from the digestive system into the bloodstream. It also secretes substances. The pancreas, for example, is critical in the production of digestive enzymes that help chemically break down macronutrients that pass from the stomach into the small intestine, and this is the pancreas' role as an exocrine organ, and it also is responsible for secretions of endocrine substances in its role as glandular tissue. We can look at the pancreas in special areas known as islets, which are responsible for secreting the hormones insulin and glucagon. It also absorbs substances. As we've discussed, the lining of the small intestine is critical in that process. This is how we allow the breakdown products of macronutrients to pass through the lining of the small intestine and get into the bloodstream in the case of carbohydrates and the amino acids that result from the breakdown of proteins, and gain entrance into the lacteals in the case of the triglycerides, which are eventually going to be packaged into structures called chylomicrons. Okay, we can classify epithelium by the number of cells, the number of layers of cells. In simple epithelium, we have a single layer of cell which extends from the basement membrane all the way to the apical surface. Well, stratified epithelia is going to be several layers of cells sitting atop the basement membrane, and the designation for the shape of the cells in the stratified layer is going to refer to the apical layer in order to name that particular tissue. And so this includes transitional epithelium, where the apical cell layers can change shape depending on how stretched out that tissue is. And this is related directly to the distension of the organ that the tissue lines. We find transitional epithelium primarily in the urinary system, where we see it in the ureters and the bladder and so on. Pseudostratified epithelia appears to be stratified when you look at it under the microscope, but in fact it's only one layer, and the reason that it looks as if it's more than one layer is due to the fact that certain cells are short and taller cells on either side lean over them, and so the nuclei aren't all lined up single file, and so initially if you look at it under the microscope it appears as if it's more than one layer when in fact it's not. If you look at the prefix pseudo shown here, the prefix pseudo means fake. Okay, so when you see the term pseudostratified, that indicates fake stratified. It's not really more than one layer. So this is in fact a simple epithelium. The shape of the cells is also one of the ways that we classify epithelium. Squamous cells are flat like a pancake, and cuboidal cells are going to be as tall as they are wide or deep, so they're shaped basically like a box. and columnar cells are taller than they are wide. And so what you're looking at here are just some different ways of categorizing cells. This is table 4.1 in your book. So what I would do is star this table and know this information, because it could very well show up on a PRS quiz or on an exam or a test. So make sure and know this. Let's talk a little bit about the functional characteristics of epithelial tissue. Simple epithelia, because it's such a tiny barrier, allows diffusion, and so gases and liquids can very easily diffuse through these layers, provided there are transport mechanisms for that to happen. And again, diffusion is based on the presence of a concentration gradient between, in this case, one side of the epithelial cell layer and the other side. We find this, for instance, in the alveoli of the lungs, which are lined with simple squamous epithelia and allow the diffusion of oxygen into the bloodstream, while at the same time carbon dioxide diffuses out. These tissues also allow for a significant amount of secretion and absorption to take place. Stratified epithelia is more structurally sound, so it provides protection against mechanical stress, such as abrasion, while squamous tissue can allow diffusion and also sometimes act as a filter. An example of the cells that do that are also found in the kidney, where we find very specialized epithelial cells called podocytes that wrap themselves around the endothelial cells of the capillaries and act as another component of the filter between the blood that comes into the kidney and the filtrate that ends up in the kidney tubules. Cuboidal and columnar epithelia are important in secretion and absorption. They may also be associated with goblet cells, which make mucus, and of course mucus has a lot of different functions. It can serve as a lubricant, and it can also serve as a method of trapping contaminants so that they don't end up inside the body, such as in its role in the upper respiratory system. Now, remember that we said that epithelial cells tend to have a free surface, a lateral surface, and a basal surface. If we look at the free surface, if the surface is smooth, the purpose is primarily to reduce friction, and this results in less physical stress, which reduces irritation, inflammation, and scarring. Free surfaces can also be populated by microvilli, which are designed to increase the surface area of the apical portion of the cell so that we can do a lot of absorption or secretion. We can think of surface area basically as bench space that allows us to perform a particular task for the body. And so the more bench space we have, the more work that we can do. Example of a place where we find microvilli on the apical surface of epithelia is in the lining of the small intestine, where we find it, for instance, in the duodenum, the jejunum, and the ileum. Stereocilia are specialized projections. These are elongated microvilli that can be designed for sensation in the inner ear. These stereocilia can respond in the cochlea to force that's produced by sound waves, and in the vestibular apparatus and in the semicircular ducts to gravity and movement. And so this is one of the ways that we're able to turn the stimulus known as sound into electrical information that the brain can understand through the 8th cranial nerve. And this is also how we're able to understand how we're moving through space as a result of, again, information relayed along the vestibular branch of the 8th cranial nerve to the brain. It tells us you're moving left or right or back or forth or around and around. There's also stereocilia that are designed primarily for absorption. We find these, for instance, in the vas deferens and the epididymis. These are components of the tube that connect the seminiferous tubules, which is the birthplace of sperm cells, to the urethra. And their role in the vas deferens and the epididymis is to absorb large amounts of fluid that produce a current that allow the sperm cells, which are not yet capable of using their flagella, to move through the male reproductive tract until the time comes for them to become part of the ejaculate, at which point smooth muscle contractions will mix them with secretions from the prostate, the bulbourethral gland, and the seminal vesicle. Uh, this will serve to activate the sperm and then will allow them the ability to swim, because it's going to be a long journey from the tip of the penis ultimately to the egg that's been released from the ovary. Cilia move materials across free surfaces. Again, examples of places where we find cilia in the human body include the upper respiratory tract, where the cilia are there to propel mucus, which can trap particulates, up towards the oral cavity, where we can either spit that mucus out or we can swallow it. In any case, the particulate and pathogen-infested mucus doesn't end up in the lungs, where it could cause more damage. And then we have folds that can exist transiently in transitional epithelium that allow the organ to change shape. And we find this, for instance, in structures that make up the urinary system, such as in the bladder and in the urethra, where when the organ doesn't contain a lot of fluid, the folds, the rugae, will be easy to see. And when the organ is rather full with fluid, the rugae will disappear as it begins to expand in volume. So let's start with our first epithelial tissue. This is simple squamous epithelium. It's a single layer of flattened cells, and we find this in the lining of blood and lymphatic vessels, where it's called endothelial tissue. We find this in small ducts and in alveoli of the lungs, where gas exchange takes place, and in the loop of Henle in the kidney tubules. This is an important structure in the kidney where we control the fluid content of the urine and, of course, the water balance of the body. We also find it in the lining of serous membranes, such as in the mesothelium, and in the inner surface of the eardrum. It functions in diffusion, filtration, protection against friction, secretion, and absorption. And you can see it's basically a diagram of the squamous epithelial tissue here, and this is an actual section taken through a Bowman's capsule. And you can see here the simple squamous epithelia that line the inner surface of the Bowman's capsule, and then these are the podocytes that are surrounding the endothelial cells of the capillaries of the glomeruli. And this right here is the intracapsular space where the filtrate is produced as the blood is filtered in an effort to clean it. Simple cuboidal epithelium is found in kidney tubules, glands in their ducts, and in the choroid plexus of the brain. It also lines the terminal bronchioles of the lungs and the surface of the ovaries, where it's designed again to protect the reproductive structure from potential infection and injury and insult. The choroid plexus of the brain is an interesting structure. These are clusters of blood vessels that are found in hollow spaces that exist inside the brain, and what the choroid plexus does is it serves as a point of production of cerebrospinal fluid, which is a filtrate of the blood that serves as protection for the brain and spinal cord, and also as a source of oxygen for the neurons that make up the brain and the spinal cord. The cerebrospinal fluid is constantly produced and reabsorbed from the circulatory system. And what does it look like? Well, it's a single layer of cube-shaped cells, and some of them may have microvilli, such as those found in the kidney tubules, or they may have cilia, such as those found in the terminal bronchioles of the lungs. And the terminal bronchioles of the lungs basically mark the end of what we call the conducting division of the respiratory system, which is basically a big fancy pipe that gets air down into the lungs. The next structure begins those structures of the respiratory system that are responsible for gas exchange, and so we call those structures members of the respiratory division. And so what does simple cuboidal epithelium do? It's involved in secretion and absorption in the kidney, secretion in glands and in the choroid plexus, and in the movement of mucus out of the terminal bronchioles by ciliated cells. Again, with the idea that the mucus serves as a way to trap particulates and pathogens so that they don't end up in the delicate tissue of the lungs where they could produce massive amounts of damage. And again, what you're looking at here is simple cuboidal epithelia taken from kidney tubules, and you can see here the lumen of the kidney tubule and the epithelial layer surrounding it. So the only way that substances can move across this layer is through some form of active transport or passive transport. Okay, simple columnar epithelium is found in glands and ducts in the bronchioles of the lungs. It's also found in the eustachian tubes, which is the tube that connects the nasopharynx to the middle ear and serves to equilibrate pressure on the medial and lateral side of the eardrum so that you can hear. It's also found in the uterus and uterine tubes, which are, of course, structures of the female reproductive system, in the stomach, the intestines and gallbladder, and the bile ducts, as well as in the ventricles of the brain. It's a single layer of tall, narrow cells, and some are going to have cilia, such as those found in the bronchioles of the lungs, eustachian tubes, uterine tubes, and the uterus, or microvilli, such as those that are found in the intestine. If we look at the microvilli on the apical surface of the simple columnar epithelium in the intestine, those are designed, again, to increase surface area so that we can more easily absorb the chemically digested macronutrients into the bloodstream or into the lacteals of the lymphatic system. And their role in the bronchioles of the lungs and in the eustachian tubes is to simply drive away particulate-infested mucus so that in the case of the bronchioles of the lungs, it doesn't end up in the alveoli of the lungs where it would cause irritation, inflammation, and damage. And in the case of the eustachian tube, it's meant to keep infected mucus out of the middle ear so that we can avoid the condition known as otitis media or earache. In the uterine tubes and in the uterus, it's designed to help propel the egg from the point of ovulation, where it leaves the ovary, to the uterus, where if it's fertilized, it'll develop over the succeeding 3 trimesters from blastocyst to embryo and ultimately to fetus and then neonate. What does it do? Well, it provides movement for particles out of the bronchioles by ciliated cells. It aids in the movement of oocytes through the uterine tubes by those cells, where they basically create a current so that the egg can eventually find its destination. And it's involved in secretion by glands of the stomach and the intestine. Stomach, of course, generating gastric juice made up of hydrochloric acid and pepsin, among other things, as well as some alkaline mucus to protect the stomach lining. And in the intestine, we produce mucus to help lubricate the food and protect the lining of the GI. And secretion of enzymes that help break down macronutrients in the intestine is also critically important as well. And of course, it's involved in absorption in the intestine. One of the purposes of microvilli is to increase that surface area so that we can more easily move the amino acids and the monosaccharides into the bloodstream, and also move the breakdown products of triglycerides in the form of chylomicrons into the lacteals, where they become part of the lymphatic system. Okay, stratified squamous epithelia is found in the mouth, the throat, the larynx, the esophagus, anus, vagina, the inferior urethra, and the cornea. If it's keratinized, it's going to be rather dry, and it's going to provide an environment that is going to inhibit bacterial growth, and we find this primarily in the skin. It's made up of multiple layers of flattened cells that are cuboidal in the basal layer, and they flatten as they get towards the surface. Hence, here we see an example of how the entire layer is named for the cells that are near the apical surface. In the keratinized upper layer, the cells are dead, and in most of the other surface cells, we find retention of the nucleus and the cytoplasm, such as in the mucosa that lines the mouth, the inside of your cheek, and portions of the oral cavity. Okay, so a difference here, right? If we look at the oral cavity, the reason that we have this moist, slick surface, but we have these many layers of cells, is the stratified nature allows it to stand up to mechanical stress and strain, and as a result, we're less likely to have irritation, inflammation, and infection. But due to its to its moist, lubricated surface, it's very easy for us to manipulate the food that we eat. In the case of the skin, what we want is to create a hostile environment for microbial growth, and so the fact that the skin is dry tends to inhibit bacterial growth. Also, the fact that the outer layers of the skin are continuously shed, they don't allow bacterial growth to set up to an extensive degree and penetrate these protective outer layers. So again, a couple of structural modifications that serve the body rather well. And what does it do? Well, it protects against abrasion and mechanical stress, against caustic chemicals, and against water loss and infection. That's the other thing that we have to acknowledge when we look at stratified squamous epithelium, is that it's like our body's wetsuit. The only way that fluid should enter the body is through food and water ingested through the oral cavity. If we didn't have this waterproofing function that's provided by the stratified squamous epithelia that lines, of course, the outer surfaces of our body and portions of the digestive system, what would happen is that every time it rained or we took a shower or a bath, we would take up massive amounts of water, and that would disturb our fluid balance, and of course that would play havoc with homeostasis. Stratified cuboidal epithelium is found in sweat gland ducts, ovarian follicular cells, and salivary gland ducts. It's made up of multiple layers of cube-shaped cells, and its function is in secretion and absorption and protection against infection. Okay, and so you can see examples here looking at tissue taken from the salivary glands. Of course, the saliva is mostly water. There are some mucus and electrolytes and some antibacterial components to it. But again, it's designed to lubricate the food to begin the process of chemical digestion, and also to provide a medium in which chemical reactions can happen, right? Liquid, of course, is going to be conducive to reactants colliding together and producing products, of course, with the help of the protein catalysts known as enzymes. And two of the enzymes we find in salivary gland secretions are salivary amylase and salivary lipase. The amylase breaks down the complex carbohydrate known as starch, and the lipase breaks down the triglycerides. Stratified columnar epithelium is found in the mammary gland ducts, the larynx, and portions of the male urethra. These are multiple layers of cells with tall, thin cells resting on layers of more cuboidal cells, and the apical layer is ciliated in locations such as the larynx. And we've already talked about the fact that the cilia help to propel secretions up towards the oral cavity so that pathogen and particulate infected mucus doesn't end up in the lungs. And so it functions primarily in protection and secretion. So we're seeing a theme here. In areas of the body where we're likely to encounter significant amounts of mechanical stress, we tend to favor stratified tissues, whereas in parts of the body that are going to see a lot less shear force and stress force, we tend to favor simple epithelial tissues. Pseudostratified columnar epithelium is found in the lining of the nasal cavity and sinuses and in the eustachian tubes, also in the pharynx, the trachea, and the bronchi of the lungs. All the cells are going to reach the basement membrane. They appear stratified under the microscope because the nuclei are at various levels, but as we pointed out earlier, in fact, what we have here is a simple epithelium because some cells are short and their taller neighboring cells tend to lean over them, and as a result it looks under a microscope as if the nuclei are at different levels and as if there is stratification, when in fact there is not. These cells are almost always ciliated, and they tend to be associated with mucus-producing cells known as goblet cells. And what do? Well, they synthesize and secrete mucus onto the free surface. The purpose of mucus in the respiratory system is, again, to trap particulates and pathogens so they don't end up in the lungs. And they move this fluid with the foreign particles over the free surface and essentially away from, in this case, the delicate simple squamous epithelia that make up the alveoli in the respiratory division of the respiratory system housed in the lungs. Transitional epithelium is highlighted by its ability to change its shape. We find it mostly in the urinary system, where we see it in the bladders, the ureters, and the superior urethra. It's a stratified tissue, and the cells change shape depending on how stretched out the organ is, basically how full of fluid it is. It accommodates fluctuations in the volume of fluid in an organ or in a tube, and it protects against the caustic effects of urine. So it provides, again, um, mechanical resistance to force, but it also has the ability to stretch so that it accommodate fluid as it fills up. And in addition to that, of course, being epithelial tissue, it's going to have nice tight seals between the cells so that we keep the toxic urine confined to the lumen of the ureters or the bladder or the urethra, and we don't allow that fluid to leak into other cavities of the body. And so here you can see an example of this tissue taken from the bladder and from the ureters. And you can see how it looks when it's stretched out and how it looks when the tissue is not distended. And so this is just a chart showing you the function and location of different epithelial tissues. Make sure and know this material as it will show up on an assessment. The next thing we want to talk about are junctions between cells. This is a critical type of structure to understand, type of phenomenon to understand, because cells need to be attached to each other for purposes of withstanding physical force, for the purposes of communicating with other cells, and for the purpose of communicating even with information in the extracellular matrix, and also as a way to restrict the movement of substances between cells so that they can only move through the cell in order to go from one compartment to another. And we'll see how these different cell junctions accomplish each of these tasks. Where do we find these connections? They're generally on the lateral and the basal surfaces of the cell. The basal surface is going to be that portion that's connected to the underlying tissue or extracellular matrix, and the lateral surface is going to be that portion that has neighboring epithelial cells on either side of it. It's important in that it forms a permeability layer, it binds the cells together, and it provides a mechanism for intracellular communication. And there are several different types of cell connections. There are desmosomes, tight junctions, and gap junctions, and we'll dissect what each of these do next. What you're looking at here is simply a diagram showing you graphically where these types of junctions are likely to be found. So let's talk first about desmosomes. These are little disc-shaped regions of cell membrane, and they're found in areas that are subject to stress. They contain adhesive glycoproteins as well as intermediate protein filaments that extend into the cytoplasm of the cell, and we often find them in stratified squamous epithelia in the skin, where they form a waterproofing barrier as well as a method of resistance to physical force. Hemidesmosomes are so named because they have the appearance of half a desmosome. They have an attachment to the basement membrane on the extracellular side, and of course they attach to cytoskeletal components on the cytoplasmic side. And it's thought that hemidesmosomes may also serve a communication role, in that the attachment of cells to basement membrane can sometimes serve to direct cell fate. We believe, for instance, that in some cases adult stem cells are signaled to remain stem cells as a result of their attachment via hemidesmosomes to this underlying extracellular matrix, whereas the other daughter cell that divides when a stem cell reproduces, losing those connections to hemidesmosomes, is free to differentiate. Tight junctions hold cells together and form a permeability barrier, and there are different types of tight junctions that we find. Zona adherens are found between adjacent cells, where we see sort of a weak glue-like effect that holds the cells together. An example of a tissue that shows this are the simple squamous epithelia in the alveoli. A zona occludens is a permeability barrier. We find this, for instance, in the stomach and the urinary bladder, and the purpose here is not to allow toxic chemicals to pass between the cells and get into other body compartments. So in a sense, they act in a similar fashion to desmosomes in that they provide this tight junction that generally doesn't allow stuff to pass between cells. Gap junctions are different. Gap junctions allow cell-cell communication, and they're essentially channels between cells that allow molecules to diffuse. Ions and small molecules can easily pass from cell to cell, and they help coordinate the function of organs such as the heart and smooth muscle, where one of the ways that we're able to move an electrical signal across a tissue surface is through the rapid movement of ions through these tiny gap junctions. Now, a gland is a very special type of tissue. Its job is to generate a product inside the cell, which is then secreted via active transport into the external environment, whether it be directly into the bloodstream or into some other chamber or lumen. And they're basically epithelial tissue with a network of connective tissue holding them together. Two types of glands are formed by the infolding of epithelium. There are endocrine glands that do not contain a duct. So what they do is they generate their products inside the cell and then actively transport them directly into the bloodstream. Examples of endocrine glands are found primarily in the endocrine system, where chemical signals known as hormones are produced inside these cells, secreted into the bloodstream, and then these signals are carried around the body by the beating action of the heart, where they find their target tissue. Those target tissues are going to contain receptors for that particular product. When the ligand binds to the receptor, it will elicit some sort of response in the target tissue, and then as a result of that will create conditions that will feed back to the original endocrine gland, indicating that secretion of that chemical signal is no longer necessary. Exocrine glands are different. They have a duct or a hose that allows them contact with an open surface or a lumen through which their product is secreted. So what happens here is that in exocrine glands we generate some sort of a product inside the cell. We then secrete it into the extracellular environment, which is connected by a duct to some other chamber, and it's through that duct that the product moves. And there's different types of methods of exocrine secretion: apocrine, merocrine, and holocrine, and we'll get to those in just a minute. Exocrine glands are classified either by their structure or by their method of secretion. If we classify them by structure, we have some that are unicellular. We find these exemplified by goblet cells, which of course secrete mucus. We found examples of those in the ciliated pseudostratified columnar epithelium of the upper respiratory system. And then we find multicellular glands. Examples of these would be thyroid glands, adrenal glands of the endocrine system, sudiferous and sebaceous glands, which are of course exocrine organs. So let's take a look at some of these multicellular exocrine glands. They're classified on the basis of the types of ducts and the mode of secretion. So let's look at the types of ducts. They're simple if they have few ducts with very few or no branches. And if they're compound, we're going to have ducts with many branches. If the ducts end in tubules or sac-like structures, we call these acini. Examples of organs that contain acini are the secretory cells of the pancreas. If the ducts end in simple sacs, they're called alveoli, and we find these, for instance, in the lung tissue where we engage in gas exchange. And so you can see examples of each of these here. You've got examples of simple glands, ducts with very few branches, okay? And then you've got compound glands shown down here, ducts with many branches coming off each, okay? Now, the method of secretion is also another way to classify these different glands. A merocrine gland essentially generates its compound inside the cell and then actively transports them via exocytosis or some other type of active transport to the apical surface, where they then move either directly into the bloodstream, or in the case of an exocrine gland, into a chamber and into a duct, and then onto a free surface or into a lumen. Examples of merocrine glands include the salivary glands and exocrine glands in the pancreas. Apicrine glands are different in that their product is actually a component of the cell. They generate a product inside the cell that's packaged into a vesicle. That component then pinches off with part of the cell cytoplasm, and it itself becomes the secretory product. An example of an apicrine gland would be the mammary glands, and their product, of course, is milk to nourish the infant. And then holocrine glands are actually responsible for having the entire cell itself be the secretion. So what happens here is that the cell will divide. The cell that ends up at the apical surface will actually pinch off from the parent cell and will become the secretion. So it'll actually die as it moves through the duct and onto the free surface or into the new lumen. And an example of a gland that does this would be the sebaceous gland, whose secretion would be the oils that produce an antibacterial environment on the skin and also lubricate the hair shaft. So again, a couple of interesting functions for those secretions. Now, connective tissue is a very different tissue type. You could think of connective tissue basically as the body's glue, but it has a lot of different functions. What differentiates connective tissue from epithelial tissue is the fact that, in general, when you look at at connective tissue, what you see are very few cells and a lot of extracellular stuff. And what generally defines connective tissue is the extracellular components that it makes and that it maintains. Connective tissue is found in every organ, and it's made up of cells separated by extracellular matrix, and there's a lot of different kinds of connective tissue that perform a variety of functions, which are listed here. So again, table 4.6 in your book, star this and know this material. So let's look at some of the functions of connective tissue. They enclose organs as a capsule, and they separate organs into layers. They connect tissues to each other. Examples of these types of connective tissue are tendons and ligaments. Remember that tendons connect muscle to bone, while ligaments connect bone to bone. They provide support and movement. An example here would be the skeletal system itself, the bones. They provide energy storage as well as cushioning and protection in the form of adipose tissue, which we know better as fat. This also provides a lot of insulation for the body so that we can help to regulate our body temperature. And of course, they're also involved in transport. Examples of liquid connective tissue include blood and lymph. Blood, of course, moves throughout the body by the beating action of the heart, as well as being assisted by skeletal muscle activity, while lymph is entirely dependent on skeletal and smooth muscle activity for movement. I should also mention that there is some smooth muscle activity that kind of assists blood flow as well. Okay, if we look at some of the other roles for connective tissue, it's also important in protection. The cells of the immune system are a component of the formed elements of blood, which keep us pathogen free. But we can also think of protection in another way. We can think of physical protection. If you look, for instance, at adipose tissue, this cushions and protects organs. If you look at components of the skeletal system, they protect a lot of our vital organs. The brain is protected by the flat bones of the skull, and the spinal cord and the brainstem are protected by the vertebral column. Okay, so protection, another very important function of connective tissue. So let's look at some of the different cells of connective tissue, and here we have to keep our eye open for specific terms. These are specialized cells that produce and or maintain the extracellular matrix. So some of these word suffixes that serve to describe their job, their role, are blasts, which actually create the matrix. An example of a cell that has blast in its name is an osteoblast. This is a cell that produces the mineralized matrix of bone, so it contributes to an increase in bone density. Cytes maintain the matrix. Examples of cells with cyte in the name are osteocytes, which help maintain the bone matrix, and chondrocytes, which help maintain cartilage. And then clasts, which are involved in breaking the matrix down for the purpose of remodeling. An example of a cell with clast in its name would be the osteoclast. So what are some other connective tissue cells that we can delve into a little bit more deeply and describe their role? Well, adipocytes are common under the skin, where they provide padding and protection, thermal regulation, and a form of energy storage. They're common in the dermis of the skin, but they're rare in other types of connective tissue, such as cartilage. Mast cells are common beneath membranes and along small blood vessels. They can release heparin, histamine, and proteolytic enzymes in response to injury. Now, you might not know what these secretions do, what their roles are. Let's look at each one of them in a little bit more detail. Heparin is an anticoagulant, and it's thought that the role of heparin in dealing with injury is to allow movement of white blood cells through the injury and towards the site of the injury, where they can do battle potentially with pathogens, and then ultimately serve as a way for the tissue rebuilding cells to engage now in tissue repair. It's also important in that it promotes blood flow to the area, and of course with blood come the cells that battle the pathogens. With blood also comes the buildup of pressure that produces pain, which we associate with inflammation, and it also increases the temperature near the injury, and this is thought to stack the deck metabolically in favor of the white blood cells and against the pathogens. So several roles here for heparin. Histamine is basically a chemical signal that promotes all of these processes. It promotes increased permeability of blood vessels, which promotes increased blood flow. That produces the pain, the swelling, the redness, and the increase in heat that we associate with inflammation. And of course this is important because as blood comes to the area, so come the disease fighters, which will ultimately knock out the pathogen and then serve as a starting point for tissue repair. And of course proteolytic enzymes, which are important in tissue remodeling once the rebuilding phase begins. This is critical. It's also important in the breakdown of structures that are no longer important once the pathogen has been dealt with. These are clot-busting proteolytic enzymes that can remove a blood clot once it's served its purpose and the tissue's been repaired underneath it. So, interesting components that we find being generated by these mast cells. White blood cells respond to injury or infection. These are our disease fighters. Two broad classes of white blood cells are the macrophages, which simply you could think of as pathogen eaters, and what they do is they gobble up infectious organisms, chop them into little pieces, and present them on the cell surface for the purpose of alerting the immune system that we do in fact have a pathogen in town that we need to be on the lookout for. The other white blood cells are the B and the T lymphocytes, which recognize the presented antigen, and based on their ability to bind to it will then reproduce at a very rapid rate, forming an army of cells that are designed to go out and chemically attack and destroy the pathogen before it gets a chance to produce disease. The B cells are the antibody producers, and the T cells are important in what we call cell-mediated immunity, where they actually destroy infected tissue or actually show up where the pathogen is and kill it as a result of a variety of chemical attacks. Macrophages we've discussed, right? They phagocytize dead or dying cells or pathogens, providing us protection against disease. They come in two flavors, the fixed and the free macrophages. Fixed macrophages stay in their position in certain tissues and glands where they're designed to filter passing fluids in an effort to scan for the presence of pathogens or infected cells. An example of a place where you find fixed macrophages are in the lymph nodes of the body. And then wandering macrophages can move in and out of any tissue compartment via amoeboid movement. So they can enter and leave the circulatory system, they can enter and leave connective tissue at will, and what dictates when and where they move are chemicals that are given off at the site of tissue infection and injury. And then there are platelets, which are fragments of hematopoietic cells that are involved in the all-important process of blood clotting, which is critical in the phenomenon known as hemostasis, where in the event of a laceration, a puncture, or an abrasion, bleeding does not continue. What generally happens is the blood will clot, the bleeding will stop, and then tissue repair will be affected subsequently. And then we have an unusual type of connective tissue, which is undifferentiated mesenchyme. And this is a stem cell type that follows the production of the three germ layers that we discussed earlier, and they have the potential to differentiate into a variety of adult cell types. Mesenchyme is a derivative of mesoderm. Okay, so it's generally got a more restricted fate than simply mesodermal cells alone. Okay, extracellular matrix is what connective tissue makes, and it's what really defines its job in the human body. It's made up of generally combinations of proteins and sugars. The collagen is one example. It's one of the most common extracellular matrix proteins in the body. It's strong, flexible, and inelastic. We find it as a major component of bone. We also find it as a major component of loose connective tissue proper, as well as tendons and ligaments. This is very tough stuff. Reticular connective tissue fills the spaces between tissues and organs. It's fine, collagenous, and it forms branching networks. Whenever you see the term reticular, think web-like. Okay, so if you look at it under a microscope, it'll have fibers running through it that look a lot like a spider's web. And then elastic connective tissue is stretchable. It can turn to its original shape after it's been stretched or compressed, and it contains elastin, which has a structure somewhat like a spring that allows it to bounce back after compression or stretching force is applied. Other matrix molecules that we need to touch on include ground substance. Ground substance is made up of hyaluronic acid, which is a polysaccharide. It serves as a lubricant. Examples of places where straight hyaluronic acid is found is in the vitreous humor of the eye, which is the clear gel that sits behind the lens and gives the eyeball shape, and also helps to pressure seal the retina against the eyeball so that you have a properly functioning visual system. Proteoglycans are made up of protein and polysaccharide. The protein part attaches to the hyaluronic acid, and this traps lots and lots of water. And then there are adhesive molecules that hold the proteoglycan aggregates together. Chondronectin is found in cartilage, osteonectin in bone, and fibronectin in fibrous connective tissue. And essentially what this does, it serves as a glue, okay, and it also serves as a barrier to infection. And it serves an insulating function and a protective function. You may have heard, for instance, of people that sometimes get skin infections called staph infections, where what happens as a result of a laceration or a puncture on the skin is that we create a port of entry, and there are types of pathogenic bacteria, pathogenic staph, that produce enzymes that can actually break down components of this ground substance, and that can serve to spread the infection. And one of the enzymes that's implicated in this is an enzyme called hyaluronidase, which attacks the chemical bonds that are found in hyaluronic acid. And so you can see where, as we can manage to burrow our way through this connective tissue layer, the pathogen could have access to the blood, where once bacteria get into the blood, that produces a condition known as septicemia, very dangerous, health-threatening condition. And this is just a cartoon showing you what some of these extracellular matrix proteins look like. You can see here collagen fibrils that are arranged in parallel bundles. Again, this is very tough stuff, resists stretching and compression, is very good at attaching things to things, muscle to bone and bone to bone, such as is found in tendons and ligaments. And here you can see the elastic fibers that we find in elastic connective tissue, which of course has the ability to stretch and bounce back. We find elastic connective tissue incidentally also as a component of distendable organs. Okay, so it would be found in association with transitional epithelium. Okay, the ability to stretch and then to contract is a very important property of these organs in order for them to carry out their function of retaining toxic fluids for ultimate expulsion from the body. And then here we can see the makeup of the proteoglycan molecule. You can pick up here the hyaluronic acid, and then attached, of course, are the chondroitin sulfite polymers. So it looks a little bit like a pipe cleaner arrangement with these molecules, but again, the major job is to serve as a barrier to infection, a water reservoir, and also has a glue-like or an adhesive property. Okay, so we'll pick this up with the next podcast. I will see everybody in class. Again, make sure to review this material as it will show up on the PRS quiz. And I'll see everybody later.